already start doing some of this. And I need to make it clear that in one hand, it's quite good because I didn't design the SGs that you used this morning. I'm not exactly aware of the system that you used this morning. And still, I was able to go back and design the SGs and figure out how you clone them and how you are going to use them. It's the beauty of molecular biology. I don't even need to know exactly what you are doing, and I can still use these tools. So we are going to talk a little bit, and please interrupt me if you have questions, and if I'm going too fast or too slow. Basically show you how we design SGs and how we clone the SGs. And by cloning, I mean we take this piece of sequence of DNA and we put them in a place in which we can actually work with and express them and all of that. And you were working in bacteria, but tomorrow morning, we are also going to do an example in which we are going to do in vitro. So you are doing this in bacteria, but tomorrow we are also going to do this in vitro.
Fabio, the idea, yes, the idea, they received the zebrafish gene that we'll see tomorrow, but the idea is to build something that they can do with the Streptomycetes genes afterwards.
Yeah.
And that will be the challenge, actually.
Okay, so my approach to this was actually trying to make it not a challenge. And that's why, as I told you before, the main constraints when you want to design SGs are these. So you need to find your target. It can be a coding region that codes a protein, or it can be a known coding sequence. It can be a promoter, it can be an enhancer, it can be whatever. The constraint is often the PAM. The cas9 that we are using is the SP cas9, the traditional one, the one that recognizes the NGG. Okay, so we are only able to find targets that are right next to this NGG. So we can go by eye to the genome and find all the NGGs and select 20 nucleotides upstream. Okay, we can do that. We don't do that. We don't do that because we need to be aware of what is the on-target efficiency. So how efficiently will the Cas9 be able to go to this place and cut the DNA? And the sequence of the spacer of the target impacts this. And we also need to be aware of the off-targets because we don't want any off-targets. If we want to cut a region of the DNA, we want to be sure that we are only cutting that region of DNA. Therefore, we don't do this by eye. We use tools, namely online tools, to select the SGs. And basically, the only thing that we do as biologists or people that are trying to design SGs is to pick and choose the sgRNAs that we want to use. Generally, these online tools, they are species specific, or you may choose the species that you are working with because there may be specificities to the species, but mainly because if you want to look at on-target efficiently and particularly on off-targets, of course, you need to be aware of the full genome of the species. If you are working with Streptomyces, you need to be aware of the full genome of Streptomyces to look for off-targets. When you do this, you work with these online tools, often the tool may give you two options. It may ask you for the name of the gene. And if you put the name of the gene, it will give you, generally speaking, only SGs that target the coding region, okay? And when you want to target non-coding regions, often you cannot use this strategy, so you need to go to a different tool, select the region that you want to target, and paste it on this online tool. And of course, these online tools have the advantage, like I was saying, of integrating multiple parameters and metrics that really allow us to select the sgRNAs that are predicted to be more efficient. So do you saw this already? I don't think so. I don't know if Mark has shown you this, but basically what I think was used when we were trying to design the SGs to target the genes in Streptomyces was this online tool, CRISPR. And in this tool, you just need to put here the name of the genes that you want to target. And I think these are the genes that you want to target. So what I did the other day was just place there the name of the gene, and there you have it. You have a list of all the target sequences that are amenable for CRISPR editing. In this list, you have different things. I will focusing on this gene. I selected the ACT5B or VB, but we could do this with any gene, and definitely the other is the same. Here we have the target region. In this target region, we have, you'll see that in a bit, that includes the PAM, okay? But in this table, you also have here the location of this target. This is the codes for the genes or the intergenic regions. And you have how many matches this target region actually have in the genome. If you are trying to target one specific region, you want one. It also tells you how many regions is this is any SG that you design based on this sequence going to match. And basically what it tells you is that if there is one mismatch, meaning if somehow the cas9 goes to a region that has just one nucleotide of difference, and of course this is unlikely but can happen with just one change, you know, biology is not perfect. If instead of sorry, you cannot see my pointer. If instead of having here an A, you have a T, it's not a perfect RNA DNA interaction. So it is unlikely that cas9 will bind there very efficiently, but it can still bind. It could still probably cut. So we need to make sure that we have as few of targets and basically that we don't have a lot of sequences coming up when we look at targets that could occur if we consider mismatches. In this case, this also includes just one, and this one is the original one, okay? But there are 14 other regions that will be targeted by this SG and by cas9 if we consider any two mutations, okay? Again, this is unlikely to happen, but it can happen because just two mutations is not a lot, and sometimes the cas9 can bind. So we just need to be aware of this. So when we want to get the guide sequence, the one that we actually are going to combine with the cas9, we need to take out the PAM. So this is what we call the spacer or the guide sequence. And this is the PAM, the NGG. In this case, is the AGG. So what we need to do is basically now to combine this sequence, that is our spacer, with the scaffold RNA or the transactive RNA, it depends, to form the single guide RNA. This single guide RNA will be combined with the cas9 to drive cas9 to its target location and cut. The trick here, and I hope, so do you have any questions about designing SG? Because this is what I have to tell you. Designing the SG is putting there the gene and having the list and selecting the best one, okay? What I will now try to tell you is how we go from finding this sequence online and actually making it work. To make it work, we need to fuse it to the scaffold, to the rest of the single guide RNA. We do that because, and this was sent to me by Marta, you guys basically have this vector that includes this scaffold or the transactivating CRISPR RNA under the regulation of a promoter. But this is just the scaffold, okay? It's just this part here. It does not include the spacer. What we need to do is to somehow fuse this sequence, the guide, upstream the scaffold. And we do that with the magic of molecular biology. So thankful, the person that designed this vector was clever, and it includes surrounding this region two motifs that are recognized by restriction enzymes. Restriction enzymes are nucleases that cut the DNA. They are basically found in bacteria. In many bacteria, they have evolved to have these nucleases, and we use them in molecular biology because they recognize generally 4 or 6 nucleotides that are generally palindromic sequences, and they cut the DNA. And this is very useful because we generally, what we do in a tube is that we take our DNA, in this case, the vector, we take these restriction enzymes, these nucleases, and we cut the DNA in vitro, and we end up with pieces of DNA. How do we clone? So how do we clone our spacer, or do we paste our spacer sequence, our guide sequence, upstream? So right here, upstream, the green. We are basically going to do this with a PCR. Okay, so basically a polymerase chain reaction in which we are going to amplify the DNA. The trick here is to include, so I should have included here maybe a slide with the concept of PCR. I hope you are more or less aware of it. But basically a PCR is a reaction in which we are able to amplify DNA to generate more copies of DNA using a DNA polymerase. It's an enzyme that synthesizes DNA based on a template. Our template is the vector. And to amplify the sequence, to multiply and create new copies of the sequence, we need primers. Primers are just small pieces of DNA that the DNA polymerase will use to create more copies, okay? So it basically, the DNA polymerase uses the primer to start the synthesis of DNA because it cannot start from zero. It always needs a piece of DNA. So what we are going to do is to design a forward primer. And in this forward primer, you will have a piece of DNA that is complementary to your template. In this case, to the scaffold, a piece of the scaffold is this yellow. We have our guide, and we have here the recognition sites. By using this, we are going to be able to introduce this spacer sequence here between the purple and the yellow. You will see that in a bit. You will also use, and especially because we want to use this for cloning later on, you will see in a bit, a reverse primer in which we'll use this other yellow region and this other purple region to amplify the scaffold of the single guide RNA. So basically, this is how the PCR works. This one is inverted because we always, like I said, need to work on the 5' to 3'. And basically, these arrows tells you how the polymerase will work. It will start synthesizing the DNA from this piece, from the primer up to here, and from the other side, from this sequence up to the end. The end result will be this, a piece of fragment that contains everything that we want to introduce, the scaffold, and the other piece that we wanted to introduce. Okay, so this is the final sequence of our PCR reaction. So in this piece of DNA, we basically, and this is done with just one single reaction. Okay, so we basically started with this guide, and this is something that we buy. Okay, so we synthesize, nowadays we can synthesize DNA. This would be a different talk. We can synthesize very big chunks of DNA even, and especially microbiology that has given results to many interesting data. But we basically just synthesize these very small fragments. This is 18 or 20 nucleotides. We buy them for like €3 or something. And we are just cloning them using a PCR. Okay, so we buy these fragments, this primer, we buy these fragments, this primer, and we end up with this. So a fragment that includes the scaffold and our guide. So we basically have here our single guide RNA. We also have these different sequences because now what we need to do and what we guys are doing is basically to express these guides in the bacteria, right? In the Streptomycetes. So we need to make sure we are placing this full sequence here, right downstream this promoter. And we do that again by using these restriction enzymes. And this time we cut the PCR product and we cut the vector or this plasmid, this piece of DNA. And we end up with this. So we end up with the vector, this plasmid cut, and the product cut. We have this purple area that is complementary because the sequence was the same and we cut with the same enzyme. So sorry, I don't know what happened. we basically end up with this piece of DNA that we can correctly insert in the right place. As a result, you will have this. You will have a plasmid that has the promoter driving expression of the full single guide RNA that is composed by the spacer and the transactivating RNA or the scaffold. And also in this vector, you have a promoter. I'm not sure if this vector is exactly the same. I think Marta did some modifications, but you have a promoter driving expression of the line. So by using these, you can express in other bacteria. And of course, then you need to insert this plasmid in your bacteria. And you may have done that already. Sorry, I don't know.
No, they didn't. This was all performed here in yes, Marta has some modifications in the plasmid. And this is the question. They have all have this protocol. And this was, well, this is one of the challenges. If the people want to follow exactly this explanation that you have gave now, we can send the PCR sequence or segment, this one, yes, this PCR product, and people may want to clone these by themselves in the different labs if they want. Okay, they just performed the final step of the thing, that is triggering the cas9 system inside the bacteria. Okay.
Okay. So, but basically, if tomorrow you want to design an SG to target a different region of the bacteria, that's the only thing you need to do.
I think, well, I think that they can do this, exactly this thing, because it is exactly what we want to do. They can try to repeat the thing that you are explaining.
And the only thing you need to buy is basically these two primers. You need to have this vector, and you need these two primers. Okay, and this is the thing that you need to buy to do the PCR amplification and then to do the cloning to insert the SG. So, and hopefully at the end, what you have now in the bacteria is the Cas9, the single guide RNA with your correct spacer, and you will be able to target the correct region and cut it. Okay.
Let me just do a question to Biophilia and to WAG teams. If we send you the PCR stuff and the plasmid without this insertion, could you perform this in your lab? Meaning, eventually, if we send the restriction enzymes and so on. Cas?
So your question is whether we could synthesize this?
Well, no, if we send you, because you have the plasmid with this insertion right now, in the, you don't, Cas, because you didn't receive the transformed.
We're not allowed to have it.
No, but you can perform this editing, this manipulation, meaning this manipulation of the plasmid. Please, Fábio, Fábio, põe no onde estavas, senão eu não consigo.
Oh, sorry.
So, I'm with the video shot, sorry. If we send you these primers, or actually, if we send you this PCR product and the plasmid, would you like to perform this kind of technique of insertion of the PCR sequence into the plasmid? Would you like to do it there?
I think so. Yeah, I think it would be very interesting. We just have to use the PCR to.
Well, actually, this is a simple digestion. You should receive two digestions, one digestion of the plasmid and the other one of the PCR amplification, and then try to connect the things.
Yeah, Yeah, that would be interesting because
And,
if there's no
yeah.
living organism there, we can, and then just put it on gel and we can see if it works.
And you, biophilia, would you like to do this afterwards?
Yes, very much. We have everything. Every facility is here except of course the primers and then the plasmid. So if we can get the plasmid, we are good to go.
Okay, okay. So I think this could be a challenge to do also. Okay. Please, Fabio, continue.
Okay, so I was just being asked, I will send you this presentation later, you may find it useful. I mean, this is the website in which you can basically find the sgRNAs. Just place CRISPR on Google, and I think it's the first result. I was just going to basically, so having this plasmid, then you already started the experiment to have your phenotypes in bacteria that I will not try to get into. But I just wanted to maybe go now how we design the SGs that we are going to use tomorrow. Just, you are not going maybe to do it because you are not going to edit zebrafish in your offices and in your labs. Probably is not so easy, but just to tell you basically how the principle is the same to whatever species. I also do CRISPR in human cells and the concept is exactly the same. We just use different online tools. For instance, in this case, my colleagues want us to delete a zebrafish enhancer that has this particularity. It controls its gene, it doesn't matter, it's called PTF1A, but the result of the deletion of this non-coding region is basically that the fish loses the pancreas. Okay, it's a very dramatic phenotype. You are cutting a non-coding region and you have a very dramatic phenotype. And tomorrow we are going to use, with Joanna that is here, I don't know if you can see it, say hello. We are going to cut this enhancer with the SGs that we used in vivo. But of course, we are going to do it in vitro, in a tube. To design SGs for the genome of zebrafish, the concept is exactly the same. We just use CRISPR scan, that is a different online tool. There are different tools we can use here. We can target genes, or we can decide to include our own sequence. Because we are targeting a non-coding region, what we did was to paste the enhancer sequence here. It's basically is this sequence here. We click predict guide RNAs, and this is the result. So it's something very similar to what we saw with CRISPR. It's a list of target sequences. It's data about the off-targets, and in this case, is a kind of on-target efficiency parameter, okay? These off-targets includes different aspects. Basically, it's again has something to do with how many nucleotides there are, and it's basically weighting where the nucleotides are, because that has been shown to have an impact in the likelihood or not of cas9 cutting off targets. Basically, then we have, for instance, in this case, I selected the first one because it's the most green. I think it's this one. We're going to use tomorrow. Because the way we are going to clone these SGs is going to be in a vector that uses a very specific promoter, is the T7 promoter. And this, oh, sorry, you're not seeing my browser again, my laser. We are going to clone this in vitro and express the SG using this T7 promoter. There is a bacterial promoter. And this promoter has the particularity of requiring that the beginning of the RNA, or the sequence, sorry, needs to be a GG. So what the software does it automatically is that if the first two nucleotides are not GG, it makes them GG. So you basically have only, in this case, you have 19, but you could end up with only 18 nucleotides that are homologous to your target region. But that has been shown to be more than enough to efficiently target Cas9 to cut DNA. People have shown that even 15 nucleotides is enough. So we basically have this sequence. This is the PAM here. And in this case, what we order, so it's a different system, we clone it in a different way, are primers that have, so these are the two oligonucleotides, or in this case are not primers. We are getting there in a bit. We just need to order these two pieces of DNA, and we just take our sequence. In this case, it would be this one with the GG, and we add in one hand TAGG and in the other CAA. And this overhang, so we anneal this in vitro. We basically we buy these two fragments, we put them together, and naturally, because thermodynamically this is more stable, they are going to anneal, get together basically. And in our case, this is the sequences that we bought that we are going to use tomorrow to cut DNA in vitro. It doesn't matter now. But we are going to clone it in this other vector that is called PDR274. And basically, as you can see, this vector has these two sites that are recognized by a different restriction enzyme that is called BSA1. It doesn't matter now. It's a different restriction enzyme, recognizes this place, cuts, and generates this cut. As you can see, this is complementary to this sequence that we have here, to this fragment that we have here. Therefore, we just need to ligate them, and we have these. And as a result, once again, we have basically this, that is our sequence flanked by these BSA sites here in blue, together with the guide RNA scaffold. So we just need to use a T7 DNA polymerase that will bind to the T7 promoter and express the sgRNA, and we can do this in vitro. So we just basically, there is a detail. If we want to do this in vitro, generally what we do is to cut the vector here with a different restriction enzyme. So after we cloned our SG, we cut the vector with a different restriction enzyme, in this case INT3, so that the DNA polymerase will stop DNA transcription. If not, it would basically continue endless through to the whole plasmid. So you need to cut the plasmid here. The T7 DNA polymerase will, the T7 RNA polymerase, will produce the RNA of the sequence until it hands the DNA, of course. So then we just need to mix these sgRNAs that we just synthesized with cas9 protein, in our case that we bind, that we buy. There may be some people and in some places that are able to produce their own protein, or if you are doing this in vivo, you could be using mRNA of cas9. In our case, we buy cas9 protein. So it's just, tomorrow we are going to mix the sgRNAs that we synthesize with the cas9 protein, and boom, we are able to cut our target DNA and have basically two pieces of DNA. This was done by Joanna Marques, I think, last year. Last week.
Yeah.
So, and hopefully we are going to see the same thing happening tomorrow, right?
Yeah.
So let's see if we can cut this in vitro. That's it.
Can we try to do the design of the sheet? The small synthesis in the website.
No. Do you want to talk through?